Yeah, so your brain is a really energy demanding organ. So it's only 2% of your body weight, but actually accounts for about 20% of the glucose and oxygen consumption, but it can't store large amounts of energy. So it's got this mechanism that it's developed, which we call neurovascular coupling, and that's a relationship between neurons and a blood flow to the same brain region. And what I do is I research this neurovascular coupling relationship in different diseases, because research suggests that actually this relationship's impaired in a number of brain diseases, and one of those is Alzheimer's disease. And I also look at comorbidity, because over 90% of individuals with dementia actually have another illness or disease. And so it makes us realise that Alzheimer's disease doesn't happen in a vacuum in life. So I think it's quite important that we investigate diseases together and then look at how that impacts pathology and potentially neurovascular function. And the way we assess that is we use animal models, and we use something called a sensory stimulation, so a little whisker stimulation, and the mice basically run on a little ball, and we then use light to look at changes in hemoglobin levels. And that gives us a nice idea of neurovascular function to this whisker stimulation, and then that evokes this hemodynamic response, we call it. And then what we can do is we can average this across different experiments and trials and different mouse groups, and then we can that allows us to then look at this evoked response, and we can then see if it's different between the different diseases to give us that idea of neurovascular function. And to give us an idea of neurovascular coupling, what we would then do is we'd also use electrophysiology, so electrodes, at the same time as looking at that changes in the hemoglobin levels, to get us like an overall idea of this neurovascular coupling relationship and is it impaired in these different diseases. Well, I hypothesised that the Alzheimer's group would have reduced neurovascular function, and that the mixed model would have the most reduced function. So the mixed is basically, it's an atherosclerosis disease with Alzheimer's disease, and that was the comorbidity we were looking at. But what we actually found is that Alzheimer's didn't have a reduced neurovascular function compared to the wild type group. The mixed model also didn't, but the atherosclerosis on its own did. So we were a bit perplexed as to why the atherosclerosis plus the Alzheimer's disease was similar to the wild type, and that's something that we're trying to go down more avenues now and potentially get more funding to try and look at that. But one thing we did find is that I mentioned we do this in awake animals, and obviously they move. So what we were doing is we would record when and how much they moved in response to the stimulation that we used. And from that we actually found that monitoring locomotion, or the amount the mouse moves during experiments, is really important, because it can actually impact that response, so it can make it bigger or smaller. And so that's really important to know if you're looking at different diseases, because if you're looking at diseases and one walks more than the other, it may actually make you think that there's a reduction in a response or a greater response, but that might just be due to the locomotion. So that's something that we've found from the work. And the next steps are that we are going to do some more experiments, analyse some of that neural activity, those neural activity experiments, to then get the whole idea of neurovascular coupling in these different models. And then we're also wanting to do some, it's called immunohistochemistry, so staining for different things in the brain, to then hopefully give us an idea of why some of these findings may have happened.